Hello guys and welcome to the Handsome Medicine. So today's lecture is about the nucleotides. So as I said on my Instagram, I will start the lectures on biochemistry. Maybe I will do some endocrinology at the same time. I will see about that. But let's start right away. So what is a nucleotide? A nucleotide is a nitrogenous base plus a ribose or deoxyribose sugar. It depends if it's RNA or DNA, and a phosphate. Okay, to make it simple, don't know. I will go into more details for you to understand every structures. So the nitrogenous bases are the purines and the pyrimidines. Why are they called nitrogenous bases? Because they have nitrogens in their structures. As simple as that. So what are the purines? The purines are adenine, guanine, and hypoxanthine. I will give you a way to remember that. It's a mnemonic, it's very famous. It's pure as gold on the 12 o'clock position. So the 12 o'clock position, maybe you never heard of this part because I made it up, but you will see why. So pure is purine, as adenine, gold, guanine, okay? And hypoxanthine, you just have to remember it anyway. You will see after. On the 12 o'clock position, why? So let's go into the structure and you will understand this part. So a purine is made of two rings. When you say two rings, it's purine, okay? With two nitrogen each. It means that the two rings, in the structure, there is two nitrogen in each ring, okay? As simple as you can see two here, two here. Don't count this one. This one is out of the ring. Hence the name nitrogenous base, okay? Like I said earlier. So the adenine, this is the structure of adenine. You don't have to learn it. For example, I looked at it on a book and then I draw it. I don't remember it just like that. I just remember there is two nitrogen in each ring, so that I remember. And other details that I will make you remember with me now. So there is an amine group here, you see it here. So this is adenine. So the only purine with an amine group here will be adenine. And there is no oxygen. You can see here, no oxygen, okay? So this is the way to remember adenine. You see a purine with two rings. You see a structure with two rings, with two nitrogenous base, nitrogen in each ring, and no oxygen, and an amine group. This is adenine. You can recognize it right away, okay? Then let's say you hydrolyze this adenine, and you take off this amine group, and you replace it by an oxygen from the hydrolyzation. You get hypoxanthine. Hypoxanthine, it's adenine minus this amine group with an oxygen instead, okay? And you have to know that in the cells there is like a turnover, for example the skin cells, and we do destroy the purines, for example. At the end of cells there is this turnover, and we do destroy these purines. Either we salvage them, either we degrade them into uric acid that is excreted in the urine, okay? So there is an enzyme, that's why I'm telling you all this now, because the next lecture we'll go into more details about that. So there is an enzyme, adenosine deaminase. But this is adenine. Adenosine is the nucleoside. Okay, I will explain that later. So this enzyme will help catalyze this reaction from adenine to hypoxanthine. To be more correct, from adenosine to inosine. Okay, and inosine is the nucleoside with hypoxanthine as nitrogenous base. Okay, now it seems very complicated, but I'm just explaining in general. And when you have this enzyme is defective, you have an accumulation of this adenosine in the cell, and the DITP after anyway. And this accumulation will lead to a disease which is called severe combined immunodeficiency. Why? Because this defect will affect more the lymphocytes, the B cells and the T cells, that are our immune cells that protect us against viruses and many things. So they die by apoptosis because of this accumulation, and which leads to this disease, this immunodeficiency, okay? So I will go into more details in next lecture, just for you to remember from adenine and hypoxanthine for how you go from one to the other, okay? So if you want to go from hypoxanthine to guanine, you add an amine group without hydrolyzation, without taking off any oxygen or anything, just add an amine group and you get guanine. So when you see an amine group and an oxygen, it's guanine. When you see an amine group alone, it's adenine. When you see oxygen alone, it's hypoxanthine. As simple as that. And I totally forgot to explain, but why on the 12 o'clock position? Because the oxygen in epoxanthine and guanine is on the 12 o'clock position in the ring. So that's why I added this for you to remember. So now let's talk about pyrimidines. The three pyrimidines are cytosine, uracil, thymine. A good mnemonic to remember that is CUT. CUT, C, cytosine, U, uracil, T, thymine. Okay? They are composed of one ring with two nitrogens each. Yeah, just one ring, so no need to say each, to be honest, but anyway. So in the cytosine you have an amine group and an oxygen here, beside the two nitrogen, of course. So that you got used to that now. And you do the hydrolysis of the cytosine and you take off the amine group and you get two oxygens here, and then you get uracil. As simple as that. So it's deamination, like here from adenine to epoxanthine. Then you go from uracil to thymine by a methylation. So this is the only time when you have a methylation, and it's a very important reaction that is very regulated, and you have a lot of drugs that work in this step of methylation. And the methyl group comes from N5N10 methylene tetrahydrofolate. It's vitamin B9 but a little bit changed. So it gives this thymine, okay, from uracil, from UMP to be more correct. But anyway, this is for the next lecture. If you don't understand what I'm talking about, it's totally normal, because now I'm just giving you an overview to understand what's coming next, which is very, very important. Even this is important. You have to know the structure first to understand the other reactions. Anyway, so to sum it up, cytosine, one amine, one oxygen, cytosine, two oxygen, uracil, and one methyl group with two oxygens, or just to see one methyl group and one ring and two nitrogens. This is thymine, okay? So I don't even need to see that to know the structures approximately. When you do the reaction, you can remember them, because it was very hard for me to remember them at first, and then I saw that this way you can remember them very, very easy. With this mnemonic, pure as gold on the 12 o'clock position, because on the 12 o'clock position there is an oxygen, epoxanthine, and guanine. And the difference between epoxanthine and guanine is that there is no amine group here. Here there is an amine group. That's the only difference, okay? So now let's talk about something else. Now let's talk about the sugar. Deoxyribose sugar, it's for the DNA. That's why it's called desoxyribonucleic acid. And ribose sugar is RNA. It's ribonucleic acid. Ribonucleic acid without the oxy. Okay. So it's a pentose. Pentose means there is 5 carbon in the sugar. So there is 1, 2, 3, 4, 5 in the structure. Okay. The source is the HMP shunt. It's also called the pentose phosphate pathway. And why is it called a shunt? Because when the glucose come in your body, the cell will take it up and it will phosphorylate it from glucose to glucose 6-phosphate. They will use two different enzymes that I will talk about later. It's hexokinase and glucokinase. Glucokinase in the liver, because there is a difference of Km, etc. But I will not talk about this now, okay? And this glucose 6-phosphate will go into the glycolysis pathway in the cytoplasm. But there is this shunt that will give it another fate, that will be this one, to give those ribose sugar to make nucleotide. To be more correct, it will give a phosphoribosyl pyrophosphate. It means that this is the sugar with the pyrophosphate here, means 2-phosphate, and a phosphate here. As simple as that, just to make it simple. And then it will be used to make nucleotides, okay? So, there is a case, I will talk about it very briefly now, of G6PD deficiency. Deficiency. No, that's all. And in this case, this HMP shunt doesn't work anymore, because the rate limiting enzyme for this shunt, the most important step, if you prefer, is this one of glucose 6-phosphate dehydrogenase. That's where you go from the glycolysis pathway to the HMP pentose phosphate pathway, okay? And when it's deficient, you will die otherwise. But you have just anemia, for example, because the HMP shunt will make NADP, that will generate glutathione, that is an antioxidant in the RBCs. Okay, but if you don't understand, it's normal. I will explain that in other lectures. Okay. So when this is deficient, you won't die, because your cell, they regenerate. Your intestinal cell, your epithelial cells, a lot of cells regenerate always. We call that label cells. So they need those purine, those pyrimidines to be made. So Why you don't die? Because you have another way, in the glycolysis, you have another way to make those ribose. As simple as that. So you don't die from G6PD deficiency, okay? You just have anemia. So now let's go into more details about those sugars. You have a ribose sugar with 5 carbons. It's called 1', 2', 3', 5', 4' and 5', okay? So the difference between deoxyribose and ribose sugar that will be used for here for RNA and here for DNA, okay? It's in the 2' position, you take off this oxygen. So when there is no oxygen in the 2' position, we call this a deoxyribose. And when there is the oxygen, we call it a ribose sugar. As simple as that. So now you have all the bases to understand what's coming. So what is a nucleotide? Okay, a nucleotide is a base, a nitrogenous base, a purine or pyrimidine, plus a sugar, a ribose sugar or a deoxyribose sugar, if you want to make DNA or RNA, it depends. RNA is to make proteins, DNA is to make the genes, etc. in the nucleus. Plus a phosphate. So you have here a base. So try to guess which one it is. So there is just one ring with 2 nitrogens. So this is a pyrimidine, so it's very clear. Then you have one amine group and one oxygen group. So which one is it? Don't look. It's cytosine, okay? But why is it written here cytidine? Because when it's in the nucleotide form, we change the name. But to be more correct, it's when it's into a nucleoside form. Because a nucleoside, it's a base plus a sugar, that's all, without the phosphate. So I will do this first. The bases, guanine will be guanosine in nucleoside. It means when you have just base and sugar without phosphate, it will be nucleoside. So guanine will become guanosine, the name change. Adenine will become adenosine. Cytosine will become cytidine. Uracil will become uridine, and thymine will become thymidine. How you can remember that? When you see purine, you think about ozine, and when you see pyrimidine, you think about idine, because cytosine, cytidine, uracil, uridine, thymidine. But when there is a purine, you have the ozine in the end: guanine, guanosine, adenine, adenosine. Okay. So the nucleoside is the base plus the sugar. Now we want a nucleotide. That's what will make our DNA or RNA. And those nucleotides are base plus sugar plus phosphate. So you have the cytidine, which is the nucleoside. You add a phosphate. Here just one phosphate is added, as you can see here. Only one phosphate is added. We could add other phosphate here, and you will have diphosphate, for example. Here it's cytidine monophosphate because there is one phosphate. If you add another phosphate, it's cytidine diphosphate. If you add three phosphate, it's adenosine triphosphate. And it's ATP. You heard about ATP that will give energy, etc. blah blah blah. Well, this is ATP. ATP is a nucleotide, just if you didn't know. So now you know what's a nucleotide. A nucleotide is a nitrogenous base, a ribose or deoxyribose sugar, and 1 or 3 phosphates. It depends. But if there is no phosphate, it's a nucleoside. Okay? So how the nitrogenous bases are made? For you to have the logic or how they made, I will give you like a little hint. From where the amino groups are, like when you see the nitrogen here, where they come from? They're from amino groups. So amino groups, amines, amine groups, nitrogen. Okay? Just for you to remember. So let's take an example of the purine here. The amine, the nitrogens here, they come from aspartate, this one, from glutamine, those two, and from glycine, this one. But there is an exception with glycine, because the carbon normally comes from carbon dioxide and tetrahydrofolate. I talked to you about this tetrahydrofolate earlier. In the purine, it's N10 4-methyl tetrahydrofolate. It's a different one. But it doesn't matter. I will explain we do a full lecture about tetrahydrofolates for you to understand the differences between them, okay? So, you have this carbon from tetrahydrofolates, this one from carbon dioxide, and the glycine gives a nitrogen and 2 of the carbons. This is the exception here in the purines, okay? So, why we call this N10 formyl tetrahydrofolate, for example. N10 is just the number of the nitrogen where the carbon is bound, because they give carbon. Like I said here, it gives to the methylation of uracil to thymine, the N5, N10 methylene tetrahydrofolate. Anyway, so the N10 means in the position of the nitrogen 10, it's bound to carbon. The other one it was N5, N10 methylene, for example. It means that between the nitrogen 5 and 10, there is the carbon that will be given, for example. That's from where the name comes from, okay? But I'll say it again. In the lecture about tetrahydrofolate, I will explain that in way more details. So, and now for the pyrimidine, we have the nitrogen, the carbon comes from carbamoyl phosphate. Carbamoyl phosphate, it's carbon dioxide plus glutamate plus 2 ATP that are catalyzed by non-enzyme carbamoyl phosphate synthase 2 in the cytoplasm. Okay, now it seems like Chinese, and it is for now. But next lecture we'll explain that in detail. So you have this carbamoyl phosphate, and all the rest comes from aspartates. So this is for pyrimidine. So it's like glycine. Here aspartates, it does the same thing, which it gives carbons and a nitrogen. Okay. So this is the structure. In the next lecture, I will tell you all the steps how you make purines and how you make pyrimidines, because it's even more complicated than what I did until now. Until now it was kind of simple, to be honest. So here you have nitrogenous base purine pyrimidine, pure as gold on the 12 o'clock position for you to remember the position of the oxygen, cut to remember the pyrimidine. Desoxyribose sugar, it means that on the 2' position there is no oxygen, so we call it the 2' deoxyribose sugar, deoxyribose to be more correct. And the ribose, there is the oxygen. And then to make a nucleoside, you make a base, it's a nitrogenous base plus a sugar, and the name changes from ozine for the purine and idine for the pyrimidine. Okay, when you add a phosphate, it becomes a nucleotide monophosphate. When you add 2, it's nucleotide diphosphate, and when you add 3, it's nucleotide triphosphate. Okay. So, and how the nitrogenous bases are made, it will be the main subject of next lecture. So I hope you enjoyed it. I hope you understood everything. And if you have any request or anything, you can ask me in the comments. And please hit the thumb up button and subscribe and whatever if you liked my explanation. And see you next time. Bye bye.